Good morning, brother.
Good morning, sir.
Good
morning, doctor.
How are you?
Very well, thank you.
So thank you, both of you, for accepting our invitation. I know you are having tight schedule because of some meetings. I came to know through Bhaskar Rao. Even then, you accepted that invitation. We are very much thankful to you.
No,
I
anytime,
know
sir.
the pains which you take.
Pleasure is all mine, sir. Pleasure is all mine.
I wish to appreciate your efforts for uplifting our profession of pharmacy by introducing some novel ideas, softwares, tools, so that especially our Indian community can have an updated version of various softwares, because we are at infant stage only.
Sure. Nice. Pleasure of you. Yep.
How the things are going
Yeah,
on?
things are going on good. Late midnight, just we are taking some sip of tea in order to keep ourselves, warm and everything. So I know.
Because it is midnight. Now the time is more than around 11:45. I think so.
Yeah, 11:30. Yeah.
So good morning. So I'm very pleased to read out the profile of Dr. Bhaskar Rao Raini. Dr. Bhaskar Rao Raini did B.Pharm from Annamalai University in 1989. He got his post-graduation and PhD from Jadavpur University, India. Dr. Bhaskar worked as professor in various capacities in the field of pharmaceutical sciences in the reputed Indian universities. Dr. Rao has authored several research international publications and presented in symposiums and conferences. Prior to industry, Dr. Raini has worked as scientist for a short tenure in highly renowned IICT, Hyderabad, India. Later, Dr. Rao has switched to pharma industry and contributed as R&D researcher at multiple positions from scientist level to director level. Currently, Dr. Bhaskara is holding the position of research fellow in the multinational Perigo. Prior to Perigo, he worked in Natco Pharma, Ranbaxy Labs, Westward Pharma, and Entech Pharma. We are very pleased to have you today as a speaker, sir. Sir, you can share your screen and start your presentation.
Yeah, thank you so much thank you, Sushma, for introducing me to the team, to the all the team. So good morning, and all the team members. And first of all, I would like to convey my sincere thanks to my fellow colleague Paritosh Kesing, who is really immediately for his acceptance. And also, I would like to convey my sincere also thanks to the management of Bapatla College of Pharmacy, respected principal, for taking this initiation, and all the team members, Satish and all, everybody, for this today's webinar. Thank you so much. So good morning. In the interest of time, I straight away go to the topic. It's a novel and a new. I cannot say new from the industry perspective. The topic is tools used in the pharmaceutical development industry perspective. So the basic aim is to disseminate the knowledge what is going on in the industry, pharmaceutical industry overall, and also to impart the same thing to the students of pharmacy. So the overview is first I'm going to cover the R&D stage to the commercial stage, and I'm going to list out the tools used in the pharmaceutical industry. So straight away, in order to have an overall idea about the pharmaceutical industry stages, the stage one is R&D, where we develop the product and process. Stage two is the process validation. Stage three is the commercial manufacturing. Any industry, when you take it's a NDA, IND, NDA, ANDA, biologics, or API, whatever. So this is the whole, the typical format of the industry, how it works. So as per the risk assessment stage, as per the ICH Q8, Q9, and Q10, I'm just giving the 3 stages overall. So stage one, typically when you see that what is mainly the work what we do in the R&D level, that is the materials usage, formulation development, process development, and also taking the concept of the QBD, risk-based QBD. So once you develop the product at stage one R&D, when you file for the ANDA, then comes to the next stage of the process validation. Typically in the stage one, we always, lock all the specifications, process parameters, all the specifications actually, right? So when you come to the stage two, we don't have any leverage of the changing any specifications. Only thing we need to verify the specifications of the process, how it goes well at a bigger scale up level. That is, pilot to the commercial scale up level. That is stage two. Typically at R&D stage one, we use a different tools, like a QBD, like a GEM tool, Minitab, Design Expert to evaluate the, to define the design space, right? So there we use typically factorial design, screening, optimization, extensive study, on the formulation material attributes, the specifications, product specifications. At stage two, during the verification of the process on the product, we see that how far extent the process is capable, like, process capability index by using, any like GEM tool, Minitab, SAS tools. After the completion of the stage two, when it comes to the stage three, commercial manufacturing, where typically we don't allow to make any changes, maybe in the process or product or in process controls or whatever. So completely it is locked, so the product is robust, ready for the launching, right? So in the stage three, commercial manufacturing and the post-commercial manufacturing, FDA is particularly emphasizing on the logistics, like particularly on the compliance part. In order to make it, more effective on the compliance part nowadays, because of the more number of 483s and warning letters, to see how the logistics, FDA is emphasizing on the usage of the effective tools called digital tooling system. That is, audits, trials, computer systems, validation. So, which I'm going to list out in my subsequent slides. So in the today's topic, I'm going to cover what exactly, how typical tools we use in the pharmaceutical industry. Maybe it is a brand industry or generic industry, Excel, SharePoint, Microsoft MS project management and timelines, Microsoft Teams, SAP, TrackWise, Documentum, LIMS, Laboratory Information Management System, Minitab, Design Expert, GEM tool, SAS, Empower tool, GastroPlus, IMS database, a Newport, and the last computer system validation. So Excel sheet is very typical. Anybody should know how to, for the comparative resolution profile, graphical representation, one has to well equipped on the Excel sheet. Then comes to the SharePoint. Nowadays, in every company is using, so the SharePoint is an excellent tool where you can see the, to plug in all the data, information, like, specifications, the scheduling system, the email attachments. So wherever you are in the globe, you can see in the SharePoint. Likewise, the next one is Microsoft MS projects. Microsoft MS project is typically the scheduling system in all the departments. Typically, when you see in the, like, R&D, analytical, quality assurance, regulatory, supply chain, warehouse, these are the typical departments in the pharmaceutical industry. So Microsoft MS projects, which is really an excellent tool to depict the stage-wise, the timelines where we are. For example, any given project has got its own, timelines. We need to complete within 1 or 2 years, right? So, for example, R&D development, how typically it takes, So, as per the monthly and weekly wise, we can depict, the scheduling and tracking, the current status, and everything will be captured in the MS Microsoft MS project tools. The next one is Microsoft Teams is also an excellent tool where we can see the framework systems, specifications, then one-on-one conference calls, then Zoom meetings and everything. You can also is one of the best tool called Microsoft Teams. So the next one is SAP. SAP tool is a very significant and excellent tool where one has to know at least fundamentals, right? For example, when you're working as a technical person in R&D, you should know, okay, you wanted to purchase any raw material, what is the raw materials item code, creation of the raw material item code, purchase a requisition to make a purchase order. So all these things can be captured, we can work on the SAP. Likewise, if any technical person, maybe in the R&D or maybe in the manufacturing, if you wanted to see the inventory availability in the, in the plant, you can use the SAP system. And also you can transfer the material virtually in the SAP 311 transactions. There are so many SAP codes which are very easy to, to memorize, like MMBE, ME51, ME 21. So that way you can, memorize all the SAP tools. So in order to know the inventory of all the raw materials, not only the raw materials, but also the finished product also. So these things also you may not be knowing in the academic purpose, but when you come to the industry, on the job training from the in every industry, typically they will impart so on the job training. So likewise, when you go to the track wise quality systems, typical quality is very important, and the currently ongoing seeing the compliance activities by the FDA. So track wise, where you can see that the management, for example, if you wanted to make any batch manufacturing in the validation or R&D or in the commercial manufacturing, it has to run as per the batch record specifications or in-process specifications or finished product specifications. If anything like a deviation, if you deviate the batch record, whatever you have prepared, approved by the quality assurance, then you have to make a deviations. There are planned deviations, unplanned deviations, these are typical. So then and there you have to capture all the deviations. So why the deviation has come in, what is the root cause investigation. So all these things can be captured in the track wise, the tool. It is an excellent tool. So after making all these changes, then ultimately when you wanted to make a change control, right, with all the changes, also those things will be captured. So that is the quality system. The quality system is very well captured in the track wise. Likewise, documentum, and also the next one is LIMS. LIMS is a laboratory information management system where it is a digital backbone of the analytical R&D, where all the data like the analytical reports, chromatograms, your standard testing procedures of all the the raw materials, finished products, and everything will be captured in the LIMS. It is an excellent tool. So by the way, the LIMS can be also the data whatever you captured can be the transferred and even to export to the GEM tool and all. So it is one of the best tool. Likewise, the Minitab, Design Expert, and the GEM tool. These are also these three. Typically, we work on the design of experiments as a part of risk-based approach of the DOE. So the FDA, mainly the emphasizing on the quality by design concept. It is not only the quality by design, but a risk-based quality approach, wherein you need to create a design space, with respect to the risk assessment variables. The ultimate aim is the critical quality attributes of the products. The criteria has to met as per the FDA guidelines, as per the QTPP of the R&D. So the next one is Empower HPLC software. When you wanted to develop any analytical method, system suitability, what is the runtime, the column dimensions, and the flow rate. So all these things can be captured by using a Empower software. If you wanted to run any HPLC, the essay or dissolution or content uniformity, you need to know, you have to be well acquainted with the Empower software system. But at the same time, so computer system, so every system tool should be validated, and also it has to be thoroughly well audited. So there will be audit trial will be there in order to prove that every system is working well as per the 21 CFR of the FDA. The next one is Gastro Plus Clinical. Gastro Plus is a mechanistic model inbuilt system, using the available PK, that is physiologically driven pharmacokinetic parameters, wherein you can simulate the modeling using the in vitro conditions of the resolution. You can build in a model where you can request the FDA bio waiver, and also you can develop the bio-relevant medium. So Gastro Plus is an excellent tool, where the typical generic industries, more than 40 or 50 molecules were approved based on the bio waiver approach. So the next one is IMS database, typically the marketing and business, wherein they see how many players are there in the market, a typical, any product, what is the sales volume, sales, all these things can be captured and, in the IMS database. And the Newport is also one of the best tool wherein we can see the active pharmaceutical ingredient sales and finance, and what is the sales forecast, how many number of global players are available in the market, you can see in the Newport. So all these tools can be, again, I said, it has to be well audited and validated. That's why there's a lot of demand for the computer system validation. IT connected pharma industry has got a lot of demand in the North America. So this is a Microsoft MS project. So stage-wise activities, as mentioned, stage-wise, you can depict here what is your, stage 1, stage 2, stage 3, and the current, what is the status, where we are. So in the month of November, so December, so monthly, weekly, what is the current status? So everything can be captured over here. It is an excellent tool. You go by the, to visualize the status of the current ongoing any project progress. So then comes to the SAP. As I mentioned earlier, so this is MM01 to create the material, MM02 to change the material, BIGO is the goods movement. So likewise, MMBE to check what is the inventory available in the plant, maybe in the warehouse, the commercial manufacturing, or R&D, or when you want to transfer any material from the commercial manufacturing to the GMP to the non-GMP, or you cannot transfer from the non-GMP to GMP. So here, this is the typical format of the SAP, material item code number, plant, what is the correct, they will define the plant, and what is the storage condition, what is the batch number. So this is a typical, SAP format wherein you can track and trace and see all the inventory of all the materials. Likewise, using SAP, you can also create a bill of materials, and also you can create a recipe meant for the, to execute the manufacturing of the registration batches. So there are so many, activities which you can, work on the SAP. So these activities, when you join in any pharma industry, on the job training, you'll be given. So each company, the systems, might be little different, but the basic fundamental but the basic fundamental concept is the same. So the next one is a mini tab, the jump tool, SAS and design expert. As I mentioned earlier, in the first phase of R&D phase, one has to work on the design of experiments using the factorial design, full factorial design. So as per the scientist has to know all these, the material variables. For example, microcrystalline cellulose, or for example, when you wanted to use a rate controlling polymer. So what is the target concentration you wanted to propose, and plus or minus as per the SUPAC level guidance 1 and 2, you need to vary all these levels for each and every raw material you are using in the drug formulation. So that way you can use the mini tab jump and design expert. It is an excellent tool. So as I mentioned, TrackWise is one of the best, the QMS quality management software system. It brings all your quality processes together in a single place to give you the big picture of your compliance and also operational effectiveness. TrackWise offers several industry best practice workflows and also can adapt to fit your existing processes and workflows. So mainly the TrackWise, of course, there are so many activities can be captured and it can be used by using the TrackWise tool. But mainly from my experience, for example, if any out of specifications in the R&D, like any deviations or CAPA is called as a corrective action and preventive action, and then audits, change controls, risk management, all these things can be captured, and also, it can be well addressed using the TrackWise software tool. So with this, I would like to conclude my short talk on these tools used in the pharmaceutical industry. And also you can see the resources: Coursera.org, Edureka, Khan Academy, Udemy, and also there are so many which you can see the freely available online sources for the tools used in the pharmaceutical industry. So with this, I would like to conclude my talk, followed by my colleague Partosh K. Singh, and after that we can have a question answer session. Thank you so much.
Thank you, sir, for your valuable presentation. Now, Dr. Rajalakshmi will read the profile of Partosh Singh, sir. And if the participants are having any doubts, the questions can be typed in the chat box in YouTube. Good morning to all the dignitaries and participants. I take privilege to introduce the resource person, Dr. Partosh Singh, sir. He is a delivery-oriented pharmaceutical professional in the formulation development scientist, and he has more than 13 years of industrial experience in drug product development with reputed pharmaceutical companies like Zydus Cadila, Johnson & Johnson. Sir, currently he is working with Perrigo as senior scientist in formulation development. He has extensive experience with developing intermediate and extended release drug product formulations with varying complexities involving key manufacturing process and technologies utilizing quality by design approach. He has proven record of accomplishment of delivering complex projects with more than 10 products, receiving regulatory approvals, NDA, ANDA, and PAS. SAR currently commercialized across the globe in various geographical markets. Dr. Partosh Singh has well versed with design and development of patent, non-fringing, patentable, stable, scalable, and bioequivalent solid oral dosage forms, powder for oral suspensions, and liquid suspension products. His accomplishments include designing and development of more than 23 products with increasing complexities for various regulated and semi-regulated geographical markets. Thank you, sir. On behalf of Bapatla College of Pharmacy, we welcome you, sir, for the session.
Thank you so much, ma'am. That's an elaborated introduction. Thank you so much. Uh, uh, Thank you, Dr. Bhaskar and the Bapatla College of Management. I really appreciate the opportunity provided to me to interact with this budding scientist here today. Um, Dr. Bhaskar, I have a presentation to share. So if you can just remove your presentation, I can share it. Thank you. Thank you. Uh, so as Dr. Bhaskar has really, I mean, the list is endless, but he has covered in great depth and details about the tools we currently use in the industry. And since it's a very dynamic thing, the tools keep on changing. We have every now and then we have some new tools which are available, which help us to do our job in a much better way. So being a pharmaceutical scientist related with the pharmaceutical development, uh, I would like today to emphasize why actually or what tools do actually we need, or the students, upcoming students or budding scientists needs to be aware of when they are like in their graduations or doing their post graduations, so that they once they are into this industries, they are very well equipped with those strengths. Um, so why is it so critical? Everybody knows that and realize that when once we say medicines, it's not an easy task. And especially developing the modern pharmaceutical drug, the formulation itself is highly challenging. So from the lab to market, just based on some of the statistics, once we start with almost 10,000 compounds, the screening, we end up having one product in the market. And so there are enormous number of phases. There are all lot of scientific things going on, research in very discrete and very different areas. And so we use a number of tools, and these tools been kept on changing from time to time. Uh, just this slide is to elaborate how challenging is the thing, moving from phase one to phase two, phase three, launch production, and then there are generic development. That's an altogether different thing. And all these different phases involves data generation. And once we are generating some sort of data, we are actually dealing with uh, understanding the data, how the data is generated, how we read it, how they are, all the data are should be auditable. And so I would limit my talk towards on using basically limiting that to the pharmaceutical development. And basically, if you talk in terms of phases, it's basically phase two to phase three for an NDA. And if it's generic, it starts from the start phase one. Because phase one to phase four of the launch, it's generally deals with whatever we do in for an NDA or new drug applications. But when you talk of a generic, this CQA, CPCP starts at the start of the process itself. But why we do that? Because a couple of decades ago, I have been in almost 2 decades. What we see is what has changed, that there is now a more and more emphasis on the quality-driven development of the data. Of the products, and that stems from the genesis of a concept which was the pharmaceutical quality by design, where it was believed, it was like thought that the mere testing and a lot of tasting into the products doesn't solve a problem of the quality. And Janet Woodcock in 2005 in an annual meeting emphasizes that we should now move to a greater use of technology into the pharmaceutical research as well. So they started the concept of pharmaceutical QBD. QBD stands for, everybody must be knowing, it's quality by design. And what does this mean is, It's a systematic approach to the development, which begins with the predefined objectives, which is known as quality target product profiles, QTTPs. Now, this systematic approach means what? Like it emphasizes the product and the process understanding. And what it ultimately aims to achieve is once we understand our product and the process, we should have a control strategy in place. So the ultimate aim of doing any QBD work is to have a proper understanding of a product, the process, and to have a proper control strategy in place. And this is only possible when everything is based on the sound science, and when everything would be explained or based on the proper risk management or sound science. So there were elements of the QBD where they say it's a systematic development through a systematic approach. Then we should have a predefined objectives, and that's especially for generic drug developments. And then we started defining, what each stage means or what we need to do that, because the FDA or the regulatory agencies does not specifically tell us what to do and how to do that. So it started with like, we should start the development with the end in mind. It's not like the every day we think something and then change our concepts. It's from the day one, we should know what we have to achieve ultimately. And then it translated into having a quality target profile and the critical quality attributes. And then it comes like, since we are using a lot of ingredients, the APIs, or we can say in terms of materials for the development of pharmaceuticals, it means that we need to identify what those materials means, that what are their critical attributes, what are their critical parameters. And now there's since a lot of using a lot of manufacturing strategies. So there's like, say for example, we've been knowing that there are like that compression, there's roller compaction methods, then there's high shear granulations, there are top spray granulations, there are hot metal granulations. There's so many. So a proper understanding of the processes so that we know what are the critical process parameters, and if we know our parameters, we know how to control them. And then in that process, ultimately, it all leads to in establishing our appropriate control strategies. So ultimately, it boils down to a thing which is called as the product and the process understanding. That is what I understand of what I after a lot of thinking, or actually what a QBD means. If you'll ask me, for me, it is like, if you cannot explain simply, you don't understand it. And that's what the regulatory agencies, or if you say the science-based development means, that if you can explain anything, it means that you understand that. So the important thing is now how to understand your product and the process. Is there a process behind that? Yes, there are definite processes or the tools which, when applied diligently, we can say with an utmost certainty to an extent level that yes, we do understand the product, the process, and we have a proper control strategy based on our understanding. So an important question is like, what do we mean by the well understood product or the process? So when we say that a product or the process is well understood, it simply means that we have identified and explained all the critical sources of variability. It means that we know what are the things, whether it's in the product or in the process, which actually impact our product performance. Then, based on our understanding, we should be able to manage the variability in the process itself. The last one is, we should be able to accurately and reliably predict what is the outcome of our changes. And this is the things, like if we see each and everything is like from the first step, like identifying the critical sources of variability. That's where we need some tools. Then we need to manage the process. It simply means that we need something to control the process, to observe the process. Tools are required. Then, now, if we want to accurately predict or reliably predict something, we need some tools. So going by the concepts, we understand that, this cannot be, the targets of QBD cannot be achieved by mere, scientific experimentations, but we definitely need some proper tools uh, to actually move towards something which is uh, science-based development, or you can say risk-based development. And here, so two things which I understand for pharmaceutical development, the two tools which are not only essential, which was, I say, must-haves for our pharmaceutical budding scientists is we should know what the risk assessments is and how to do a risk assessment basically. Because whatever we do, whether we analyze, whether we strategize, whether we make a plan, whether we are assessing something, there is a risk involved. So there are a number of tools available to do this risk assessments and to perform our mitigation strategies. But unfortunately, the list is a bit long. So the idea is, if we at least know few of them, which are required based on what we want to do, whether we want to identify the risk, whether we want to understand the consequences, whether we want to see the probabilities, or we want to have a level of risks, whether low, high, medium, we need to do that, or we want to just evaluate the risks. There are definite tools available, and out of them, there are a couple of them which are basically used in our pharmaceutical development, that is dosage form development. So just for an example, here is a list like how we do the risk assessment. Say, for example, it's a simple an example of a drug substance. So when we have a drug substance in place, we start the development, and we know that what our CQAs, and the critical quality attributes of the products are. Say it's like NSA, content, degradation, dissolution, at the minimum. And then we have a drug substance, and we just need to have this kind of a table, it's kind of a risk assessment table where we see which attribute of that API, or you can say a material attribute of that specific substance we are talking of, will actually impact these of the CQAs. So the list is enormous. It's the result of, it's not we get somewhere in any any book or a paper. It's just a team comprising of the pharmaceuticals, analytical scientists will sit together and then identify what risk assessments or what are those attributes of the specific materials which actually impact our defined CQAs. On the right hand side, we see a tool which is many of you must have seen before, a fishbone or a Ishikaga, to identify the cause and effect relationship. That should be like, that's very extensively used for in the pharmaceutical industry for as a risk assessment tool at the start of the process itself. Here's just an example of a product, and we see here like we are trying to analyze. So it's a say for example, it's a tablet dosage form, and we are trying to understand the ingredients, how these ingredients will actually impact the defined CQAs. So this fishbone diagrams, as Bhaskar, Dr. Bhaskar previously mentioned, the SHQ8, Q9, Q10 should be in a Bible, which should be like, it should be in, it should be crammed, it should be in the heart of every pharmaceutical scientist, because these forms the basis on which all these different concepts has been developed. Here, once we do a risk assessments, we know what are the characteristics we need to control, what are the properties we need to monitor. And then here comes, we know everything. We have in skeleton, we have a database now, which excipients we can use, what are the processes we can use. And then what we do is, every project involves experimentation. We need to do experiments to generate data. Now, there's a question like how to do that experimentation. In earlier I remember a decade back, or maybe most of the time right now itself, we used to be doing OFAT method of experimentation, wherein we keep everything constant, change one parameter or one ingredient, one concentration, and do an experimentation to see uh, what is the impact. Unfortunately, although we have some say it has solved a purpose of experimentation for so much long time. There are some very significant problems with this method of experimentation because generally we know that in pharmaceuticals there's always more than 2 to 3 ingredients involved, and with this OFAT method of experimentation we don't know what would be the interaction impacts, and that cannot be found reliably using an OFAT method of experimentation. So then it starts like, what is the tool? Again, we needed a tool to do an experimentation wherein we can study the interaction impact simultaneously. So as discussed before, we have several ways of experimentation. There's one which is called trial and error. We do once. If it hits the target, well done. It's good job done. But we don't know whether the optimum is. There is one factor at a time. We make an experiment, it's much better than trial and error. We do one experiment, we move to the next by changing. But as we have seen, we have several disadvantages here in terms that we do not have data on in the interactions. And then it came like the structured experimentations. So what does it mean by structured experimentation? Because every experimentation is a structured experimentation, whether it's a trial and error, whether it's a one factor time approach, or whether it's a DOE. So structured experimentation here refers to what's known as the design of experiments, popularly known as the DOE these days. And this is an way of, DOE is a way of effectively and efficiently exploring, the cause and effect relationships between the product and the process variables and the output, whatever we can call, sometimes called a response, sometimes called an effects. There are a number of ways to denote this terminology. As a pharmaceutical formulation development scientist, it is we can use different softwares available, to carry out it's an experimentations, because there's so many number of different designs we have to choose. The most popular these days, which is used across the industries, is a GMP software, and GMP is from the SAS. It's a well-known institute, which is I think it was launched in 1989, and it is a very good interface. But definitely it's a bit tricky to use. Before JMP, two very well used softwares for doing the experimentations were Minitab and Design Expert. I think in India, Design Expert is being used very widely, and I think its studies is there, and they routinely do what's called as trainings to use this software. So all of them, be it JMP, be it Minitab, be it Design Expert, my understanding, what I can see from here is, doesn't matter which software we use for our development, because softwares may be different, interfaces may be different, but what it involves is understanding of the concepts. And what I see a difference in a decade back and now, if we can focus a bit more on the mathematics and the statistics part during our graduation, especially, and if I'm not sure how the syllabus looks like today, but if it can be included, that would be the best, because whatever software we use, all the things remains the same. It's just that if you don't have any understanding of the concepts, because everywhere the statistics, the mathematics is involved. So GMP is more than it's a very vast, and it's not only used just for the pharmaceutical designing of the experimentations, but there is a lot of statistics and other things which it can do. Uh, Especially if we are looking for a software which is very popularly and very widely used just for the designing per se of pharmaceuticals. It's the design expert by Statis, and I think that's very popular. I know that it's this tooling thing. Tools used in industry is such a wide area. We cannot be dealt in a class or in our presentations. It's a huge. But what we thought, what my view was like, we just wanted to show or present to the people, the team here, the budding scientists, that these are the few of the things which should be must have, especially you if we are planning to have a career in the pharmaceutical development. And that's the risk assessment tools and the DOEs. Doesn't matter which softwares we're using, either JMP, Minitab, Design Expert, and if there's another, I don't know. But statistics is one of the things um, we should have an access to, and at least have a proper basic understanding of different statistical concepts, calculations, uh, and if possible, we should also be a slightly not a handicap for some coding informations languages, because nowadays the next thing which is coming into the next decade for pharmaceuticals will be coding. We can just make our own softwares, we can make our processes, if we have those informations. So this is for pharmaceutical development, the solid dosage form, liquids, or whatever it is, for developing formulations. The another one which I would like, and as Dr. Bhaskar has already covered with his presentation about, this is just how it looks like, when we talk of factorial designs, or like um, whatever, like it's half factorial, full factorial, minimum resolution runs. But my thing is, if you don't understand what does it mean to run up to 13-6 or 4 resolution experimentation, it doesn't make sense, to have a software. So it's really important to understand the statistics, the facts behind, and the softwares is. I think it's not that difficult to catch up with that. Another area which is very much growing up with leaps and bounds today in today's time is the Gastro Plus or the Simulation Plus. That's a software. That's something which is, what we understand is like predictability. It gives you, it may be a surrogate for predicting your bioequivalence, like, or clinical data. So maybe in future it might be that once we have a proper model developed based from the simulations, we even don't need to do some costly and painful clinical studies, and they are like very costly. They are time consuming, and it's a lot of resources needs to be invested for clinical studies. So the future belongs to this way of clinical science, where we will be using these softwares to properly mimic the GI physiology, and so that based on our models developed, we can actually predict the pharmacokinetics and how the drug behave inside our body. So with this, I would like to say thank you to all of you for being so patient. With this, I'm just done with my presentation. Thank you.
Thank you, sir. Thank you for your valuable information, sir. So, friends, if please ask her. Sir, can you stop the screen share, sir? Parthos, sir.
Yeah. Oh, yeah, sure. Yeah. Yeah. Thank you.
Anybody? Thank you very much, sir. Thank you very much for being with us. So you spend your valuable time for us. Thank you once again, sir.
No problem. Thank you. Anytime. Sir. Thank you. Thank you for having us here.
Thank you. Thanks. Bye, sir.
Right. Thank you. Mhm.
Certificate, not product list. Okay, sir. Thank you very much, sir. Bye, sir.
Thank you. Bye. Thank you.
So participants, next session, we are going to continue with your presentations. So after taking the break of 10 minutes, so please join according to your schedule. Thank you.